Distinguished professors, I'm Yang Xinyue. It's a great honor for me to be here, sharing our experimental results and the latest advances. The project I'm presenting is a study of the mechanisms of non-adjacent endothelial cell injury induced by pyroptosis caused via bubble contact. My report is structured into 6 sections. 1,000 years ago, our ancestors voyaged across the sea, forgoing the maritime Silk Road. In the new era, the exploration and utilization of marine resources are crucial for our sustainable development. During this process, decompression sickness, a medical concern that endangers the divers' operational safety, has drawn much attention. Arising from insufficient decompression, DCS often progresses rapidly and leads to severe consequences, making its prevention and treatment a top priority in diving medicine research. Vascular endothelial cells are located in the innermost layer of blood vessels and serve as crucial receptors and effectors for maintaining the body's homeostasis. When VEC damage occurs, it can result in increased vascular permeability, vasodilation and contraction disorders, coagulation abnormalities, and the activation of an inflammatory response. In our previous research findings, VEC damage and a systemic inflammatory response were frequently detected after diving and in animals subjected to hyperbaric exposures. And based on our observations, we demonstrated that Acin and Simvastatin can reduce the incidence of DCS via VEC protection. These results suggest that VEC damage is a key segment of inflammatory response in DCS. Pyroptosis is a novel mode of cell death in which cells are stimulated to form the NLRP3 inflammatory complex and ultimately leads to cell rupture. As a pro-inflammatory cell death mode, pyroptosis can trigger the activation of immune cells and the release of interleukin-1 beta, interleukin-18, and HMGB1, which ultimately initiates an inflammatory cascade and plays an important role in various diseases, such as diabetic vascular disease and hypertension. Based on the experimental results from scanning electron microscope, PI staining, and ELISA, we confirmed that human umbilical vein endothelial cells undergo pyroptosis after a single bubble contact. Given that VEC damage is a key segment in the inflammatory chains of DCS, we hypothesized that VEC pyroptosis plays an initiating and amplifying role in the inflammatory response of DCS. Drawing on our preliminary observations of VEC pyroptosis, we've devised a comprehensive technological pathway for further in-depth research. After successful construction of an injury model, we will clarify the pattern of non-adjacent endothelial cell injury and further explore the mechanism of the bubble-induced pyroptosis. First, we use a Shui valve connected to a syringe to mix the extracellular matrix with gas, simulating the damage caused by bubble contacting endothelial cells in the modified transwear medium. Like this, we make bubble contacts at the cells in the upper chamber and detect damage to the cells in the lower chamber, covering 2 scenarios. One is to measure the damage indicators at different times after contact, and the other is to vary the contact durations with the upper chamber cells and then measure the damage. Subsequently, we will analyze the morphological changes, interleukin-1 beta and interleukin-18 by ELISA and NLRP3 via Western blot. Ultimately, we aim to analyze the pattern of injury spread. During pyroptosis diffusion, damaged endothelial cells release HMGB1. So in the next period, the HUVECs were pretreated with pyroptosis inhibitor MCC950 for 30 minutes. And then the upper chamber was touched with multiple bubbles. by using pyroptosis inhibitors, ion channel inhibitors, and HMGB1 neutralizing antibody, we aim to analyze the mechanism of the damage caused by bubble contact. To date, we've successfully developed a cell layering culture technique and constructed a multi-bubble contact and a single bubble contact injury model. Also, the elevation of intracellular cation ions after bubble contact has been confirmed, aligning with the trend seen in PI stain diffusion. By administering different cation channel inhibitors, the intracellular cation ion fluorescence decreased, along with a reduction in cellular damage. This suggests the involvement of TRPV4, TRPC, IP3R, and SOC channels in the inward cation ion flux during pyroptosis spread. At last, the innovation of our study lies in systemic experimental design of DCS pyroptosis spread, creative construction of stratified culture technology, and innovative application based on the present techniques. Also, our research group is the first to explore the mechanism of DCS, inflammatory onset and damage spread. That concludes my report, and I sincerely welcome your comments and suggestions. Thank you.
Thank you very much for giving such an interesting presentation. Does any judges have any questions?
So thank you for your nice presentation. So I just wonder that how can you apply your finding to the use of this, to inhibit the inflammatory when they expose with bubble? How can you apply it to the real life?
Oh, yeah. So nowadays, both the military and commercial diving continues to evolve, and during this process, decompression sickness is an important medical concern that can endanger the divers' operational safety. So our research group aims to explore the pattern of injury spread and the mechanism of the bubble-induced pyroptosis, because nowadays the treatment of DCS is still limited. There is no targeted drugs for the treatment, and when the DCS happens, the divers can only undergo the hyperbaric oxygen chamber to recovery, and our research group aims to explore the mechanism so that we can find a suitable therapeutic target for protecting the endothelial cells and to alleviating the inflammatory response via the VEC protection for the treatment of the DCS. Thank you for your question.
Towards the end of your presentation, you show several genes that may be impacted by bubble injuries. Yeah. So this one. So are these the plans for further studies in the future, or did you already validated that these are the target?
Oh yeah, we've detected relevant cation ions that are involved in the spreads of pyroptosis. And further, we will conduct more research on the relevant ion channels to explore the mechanism of the pyroptosis spread. Yeah, thank you for your question.